Okay, the next thing we're going to talk about is polygenetic inheritance. You do not have to do any calculations, or I just want you to have a general understanding of what things are inherited in this manner and what it means. So, like I mentioned, some of our genes, a single gene is responsible for a single phenotype, but most of our phenotypes are the result of the expression of several different gene pairs located throughout our chromosome. So, what's going to happen is that we actually get what we refer to as phenotypic variation. And typically, this is going to be a continuous or quantitative variation between two extremes. So, examples of things that are inherited this way are skin color. So, it's kind of again a continuous variation between two extremes of very, very pale skin to very, very dark skin. Another one is height, our intelligence, and our metabolic weight. So, kind of understand those four things and how they are inherited this way. So, an example of polygenic inheritance for our skin color, again, alleles for dark skin are capital A, B, C, and are incompletely dominant for those for light skin, which are the lowercase versions. Again, you will not have to understand across or have to do anything with this unless you move on to take a genetics course. For now, I just want you to understand some of our phenotypes that are inherited this way and what it means. So here's just an example, and this is why we're not actually going to do it because it gets a little crazy. But this is our basically our two extremes of skin color here. And typically the highest amount that we have in a population of this type of variation is going to be right in the middle between the two extremes. And then we have less proportion of our population making up our two extremes, and it makes up what we refer to as like a bell-shaped curve. Okay, so in some situations, our environment that we are actually in can actually override or influence which genes we actually express. So, things like drugs or even pathogens or viruses can go in and alter our normal gene expression during embryonic development. So, one example was thalidomide. This was a morning sickness drug that was used in the 60s. And what they found is that embryos actually developed phenotypes that were not directed by their genes, but were actually directed by this morning sickness drug. So, example of phenocopies, when we refer to the term phenocopies, that means we produce these phenotypes by our environment. It wasn't actually produced genetically. So, environmental factors can also influence gene expression after birth. So, for example, if we have very poor nutrition, that can affect the growth of our body along with the growth and development of our brain. We also have childhood hormonal deficits that can lead to abnormal skeletal growth and proportions. And we mentioned that in the endocrine system and some of the hormonal effects. Okay, so genetic outcomes can be influenced by other non-traditional controls, such as things like non-DNA regulation of gene expression. So basically RNA only genes. We also have chemical groups that can attach to DNA or our histone proteins. And another thing is extranuclear DNA that we have, so mitochondrial inheritance. So we have three levels of controls that are found in the human genome. The first layer is going to be our protein coding genes. So this is actually going to involve less than 2% of our cell's DNA because most of our DNA is actually junk. So we have all of this DNA, but only about 2% of that actually codes for proteins that we're actually using. So the DNA is going to be what we consider the blueprint for protein synthesis. The second layer is what we're going to refer to as small non-coding RNAs, and these are found in non-protein coding DNA, so something like for RNA. And this makes up about 80% of our genome. The third layer are going to be epigenetic marks, and these are stored in proteins and chemical groups that bind to DNA and in a way in with the chromatin that's actually being packaged. So the first thing we're going to talk about are small non-coding RNAs. So some examples are microRNAs, small interfering RNAs, and what we call Pee-wee interacting RNAs. And the whole role or the function of these small non-coding RNAs is to reduce the expression of some genes. And they join with enzymes to identify targets that lead to form RNA enzyme complexes. So for example, we can have destruction of target mRNA by dicing it up into smaller fragments. If you've ever learned or studied about the CRISPR-Cas system, that's one method that you can use these small non-coding RNAs. We can also inhibit translation of the target mRNA and silence the gene that codes for the target mRNA, basically by going in and modifying histone proteins that help package the DNA. So they inactivate transposons. Transposons are what we call jumping genes because they can kind of jump around. And these genes that tend to replicate themselves and insert themselves into distant sites, they can either disable or hyperactivate genes wherever they're at. They play a role during development. They also help fight off viral infections, and they are being used, as I mentioned previously, as tools to treat a variety of diseases by interfering with the expression of certain genes. If we have any mutations in these small non-coding RNAs, these have been linked to cancers as well as schizophrenia. So our third layer of control was our epigenetic marks, and this is information stored in proteins and chemical groups that are bound to DNA. So we have these chemical tags that determine whether or not the DNA is going to be available for transcription, or we're going to say no, it's going to be silenced and not available. And these two important tags are acetyl groups and methyl groups. So if we have an acetyl group attached to our DNA, that means that this specific area of DNA is available for transcription and eventually translation to form the protein. If we have a methyl group bound to this particular region of DNA, that's going to silence it, which means that that portion of the DNA is not going to go through transcription or translation, and it's not going to be expressed. So epigenetic marks or not having epigenetic marks can actually predispose cells to cancerous changes. We have genomic imprinting, where certain genes are actually tagged with methyl groups during gametogenesis to help identify them as either being maternal or paternal. So these are essential for normal development and allows the embryo to express only either the mother's or the father's gene. Each new generation erases the imprinting when new gametes are produced. So examples of genomic imprinting can be seen in mutations of imprinted genes that result in pathology. Same allele can have different effects depending on which paternal chromosome was actually being expressed. So one example is deletions in chromosome 15, and that's going to result in disorders with two different symptoms. We have Prader-Willi syndrome is seen if the mutated gene inherited from the father is expressed, and we have Engelmann syndrome is seen if the mutated gene is inherited from the mother is being expressed. And depending on whether it came from the mother or the father, also is going to determine what phenotype it has. So if it was inherited from the father, the syndrome that's actually produced is going to cause moderate intellectual disability. If it's inherited from the mother and expressed, it's actually going to end up with severe intellectual disability. And the last thing we need to talk about is extranuclear inheritance. So what we're referring to here is DNA that's located in the mitochondria and not in the nucleus itself. So our mitochondria contain 37 genes of their own that we refer to as mitochondrial DNA. And the mitochondria are transmitted to the embryo by the mother because the ovum donates essentially all of the cytoplasm in the fertilized egg. So if we have errors in a mitochondrial DNA, they are linked to rare disorders, and usually these are problems associated with the function of the mitochondria. So problems associated with oxidative phosphorylation or cellular respiration, as well as some muscle and neurological problems. Now they're kind of linking it. It's not 100% sure, but they're kind of linking these errors in mitochondrial DNA to Alzheimer's and Parkinson's disease. Okay, carrier recognition. So there's two ways to identify carriers of detrimental genes. We can either use pedigrees or we could use blood tests or blood chemistry, where we're using DNA probes to detect the presence of some unexpressed recessive genes. But basically the function of a pedigree is to kind of trace the genetic traits through several generations. So a lot of times in order to figure out what genotype you are, you could go back to 2 generations to determine current generations, or vice versa. If you know your generation, you can kind of work backwards. We're not going to go into learning to read these in this class. Okay, the absolute last thing I want to talk about is gene therapy. This is a huge new field and something you may be interested in the future. And genetic energy engineering has the potential to replace defective genes. This can include defective cells that are infected with genetically engineered viruses containing a functional gene, and they basically inject corrected gene into the cell. So what they do is they use the CRISPR-Cas9 system, which is actually a bacteria's own immune system, and they modify it and engineer it so that it can target a specific sequence within our own genome. And when it goes in and it can target it, it's going to cut it, and it can cut the old copy out and insert a new copy of the corrected gene right into our DNA. So this brings up a lot of debate. Just because we can do this and basically cure any type of disorder, genetic disorder, with this system, does that mean we should do it? So again, many ethical, religious, and even societal questions of whether or not we should do this. So they actually did the first treatment was actually a SCID treatment that was successfully accomplished by this CRISPR-Cas genetic engineering.